All right, today we're diving into something that feels like it's pulled straight from a sci-fi movie. We're talking about the actual tools scientists use to edit the very code of life itself, DNA. We're going to break down exactly how these incredible molecular machines actually get the job done.
So that's really the big question, right? I mean, if DNA is the fundamental instruction manual for every living thing on the planet, how on earth can we go in and rewrite a sentence? It's one thing to read the book, but a whole other challenge to go in and be the editor.
But why even bother? Well, take a look at this. This isn't just some abstract science experiment. We can actually use genetic engineering to create bacteria that literally eat heavy metal pollution from old mines or digest toxic waste. So it has the potential to solve some really big, real-world problems. And that's exactly why understanding the how is so critical. Okay, our first tool is all about making tiny, super precise changes. Imagine changing just a single letter in a word that's thousands of pages long. It's called site-directed mutagenesis, but honestly, a much cooler way to think about it is the Trojan horse method. The whole goal is to sneak one little change into a cell's DNA without it ever noticing.
So here's how the mission works. First, scientists build a tiny piece of DNA called a primer. This primer is the Trojan horse. It's almost a perfect match for the original DNA, but it's hiding one single secret change. So when it binds, it creates this little bulge where it doesn't quite match up. Then, the cell's own copy machine, DNA polymerase, just plows right over it. And because it doesn't notice the tiny defect, it copies the whole thing, locking our secret change into a brand new strand of DNA.
So now you have the new DNA, but the old unedited stuff is still hanging around. How do you get rid of it? You bring in the cleanup crew. In this case, it's a special enzyme called DPNI. You can think of it like a highly trained assassin. It's programmed to only target the original DNA, which has this specific chemical tag on it, and it totally ignores the new version we just made.
And that's the key. DPNI just chews up all the original templates, leaving behind only our new plasmids, the ones that carry the mutation we wanted. The Trojan horse did its job, the old guards are gone, and the new code is in charge. Just like that, a perfect, tiny edit is complete.
But what if changing a single letter isn't enough? What if you want to replace an entire paragraph or a whole gene? Well, for that, you need a different tool, something for a much bigger edit. This one is called homologous recombination, but we can just call it the perfect swap.
What's so clever about this technique is that it actually hijacks one of the cell's own natural DNA repair systems. Now, I know this diagram looks a little busy, but the main idea is pretty simple. Scientists create a new piece of DNA with these homology arms on the ends. Those are the orange bits. These arms are designed to perfectly match the DNA sequences on either side of the gene we want to replace. So when this new piece gets into the cell's machinery sees those matching arms and thinks, aha, I know exactly where this goes, and it kicks off the swap.
So once the right spot is found, the new DNA and the chromosome do something really cool. They physically cross over, tangling up into this X shape you see. This is the moment right before the big exchange, when everything is lined up and ready to go.
And here it is, the final result. The cell's own machinery cuts out the old gene and stitches the new one right into its place, perfectly. And you see that part labeled resistance? That's usually an antibiotic resistance gene, and it's added in as a little bonus. It acts like a flag, so scientists can easily find the cells where the swap actually worked. Old gene out, new gene in, simple as that.
Okay, and that brings us to our third and definitely most famous tool, CRISPR-Cas9. If our first tool was a scalpel and the second was a transplant, this one is a programmable guided missile. It has totally changed the game because of its incredible precision and how, relatively speaking, easy it is to use.
The system is really a two-part team. First, you've got the Cas9 protein. This is basically a pair of molecular scissors that can cut DNA. But by itself, it's pretty useless. It has no idea where to cut. That's where its partner comes in, the guide RNA. The guide RNA is the GPS for the scissors. It's a tiny piece of RNA that scientists can program to match any DNA sequence they want to target.
So together, they form this incredible search and destroy unit. The Cas9 protein, armed with its guide RNA, just cruises along the DNA, scanning everything. When that guide RNA finds its perfect match, and I mean the exact sequence, the whole complex locks on. And then, snip, the Cas9 scissors make a clean cut right through both strands of the DNA.
So, what happens after you cut the DNA? The cell kind of freaks out and rushes to fix the break. But its emergency repair system, well, it's sloppy. It's prone to making mistakes like adding or deleting a few DNA letters by accident.
And usually, those little errors are more than enough to completely break or knock out the gene you were targeting. So it's this brilliant trick where you use a really precise cut to cause a messy but very useful disruption.
So let's pull all this together. We've looked at 3 really different ways to manipulate DNA, and it's not about one being better than the others. It's about having a full toolkit and picking the right tool for the job.
This table really sums it up, doesn't it? The Trojan horse is for that tiny surgical change of a single base pair. The swap is when you need to replace a whole chunk of DNA, an entire gene. And the guided missile, CRISPR, is your go-to when you want to target and just break a gene, to knock it out. Each one is a master of its own craft.
And here's a fascinating final thought on CRISPR. This revolutionary technology, we didn't invent it from scratch. We discovered it. It's actually a natural immune system that bacteria have been using for millions of years to fight off viruses. We just figured out how to take that ancient defense system and reprogram it for our own purposes. How cool is that? So from fine-tuning an enzyme to replacing a faulty gene or knocking out a harmful one, these tools have completely changed what's possible in biology. Which, of course, leaves us with a huge question. Now that we can actually rewrite the code of life, what story are we going to choose to write next?